slide is actually showing you cell-to-cell communication. Usually when you finish 430, most instructors, they end with the nervous system. And that's where we teach students how to learn about cellular communications, neuron to neuron. So you learn about the synapse, you learn about neurotransmitters. Here's an example of this top one is called a paracrine response. It's another form of cell-to-cell communication where it's a localized response. One cell is just secreting something that affects a nearby target cell. Paracrine, it's localized. Cellular communication, which is localized. So within the same tissue or organ. The bottom picture is showing you an autocrine response where the secreted substance affects the cell that secreted it. Secreted substance affects the cell that secreted it. So cells affecting nearby cells, cells affecting themselves. This chapter is devoted to talking about how the cells can affect distant cells, not nearby, not within the same tissue or organ, not localized. And that's endocrine. Okay? And that's a pretty fair picture of what happens. But the point is that you're using the bloodstream to get the communicating molecule to affect other cells. And those chemicals are called hormones. So let me give you the formal definition of the endocrine system. Endocrine response involves, first, endocrine glands. They synthesize chemical messengers called hormones. Endocrine glands synthesize chemical messengers called hormones. Which are secreted into the bloodstream. That should be ERS. Hormones travel to specific target organs. The hormone causes some response in the target organ. this is in paragraph form, the picture. You're seeing everything which you wrote on that slide. Important things to underline. When you study this as a student, you need to like figure out, well, what gland am I studying? And what's the name of the hormone it's secreting? And you got to know what it targets. Specific target organ. It's something far away, usually, and also, well, this is a physiology class, what response does it elicit? That's the physiology. So you got to know it all. Okay, and these travel to faraway places. The bloodstream is important because blood goes everywhere. So these are secreted in your bloodstream. They circulate everywhere. But you don't want it turning on everything. There's specific things you want to affect. So these distant target cells, you're affecting certain things, not everything. So I need to spend a lot of time unpacking hormones and get some key points across there. And we'll go through these as I progress here, but I mean, the first thing that should come across to you, because hormones use the bloodstream, there are long distance chemical signals. Okay, unlike the synapse, the neurotransmitter has to like just traverse the synapse, which is a very microscopic space. These messengers use the blood to get from, brain to kidney. They travel a far way. And so we'll talk about all these points, their chemical structure, and how they elicit responses and things. And so a good example to start with for students for hormones is this figure here. It's teaching students the effect of the hormone insulin and glucagon that regulate blood sugar. And when you study these things, you can get bogged down memorizing glands and hormones, but what is the body trying to regulate? In this example, it's blood sugar. In the fasted condition, it should be like, less than 100. So let me show what's going on here. Example, the regulation of blood glucose. Now, the circulation form of sugar is glucose. And normally it should be about, less than 100 mg per deciliter of blood. You eat a meal, pasta or apple or something, your blood sugar goes up. Eat pasta. Your digestive system breaks down the starch, the simple sugars get absorbed into the blood, increase blood glucose. And your body needs to take that nutrient molecule and it needs to do something with it. It needs to get it to the tissues that can burn it and use it. And so you have to have a mechanism to detect that. Now that organ, the pancreas, is behind the stomach. It's right here. This is your endocrine gland right there. It has a way to sense the increase in blood sugar. I was inverted to back of hyperlipid pancreas. That's your detection mechanism, and it'll secrete into the bloodstream hormones. In this case, insulin. So let me back up just a step. The first thing that got elevated in the bloodstream was glucose. Yeah, so let me symbolize that with blue dots. That got absorbed. So that was there first. And what we're saying is the pancreas can detect that elevated glucose and in response to that, secrete in the bloodstream with it insulin. about we used green squares to symbolize insulin? So they're circulating in the bloodstream together. Insulin needs to stimulate the correct tissues that can extract the glucose from the blood. Well, they give you a couple examples. They say tissue in the liver. Liver is one. Skeletal muscle is another big one. So you got the liver, and then skeletal muscle. So let's say you have some cell from the liver and the skeletal muscle. They present a receptor to insulin. In your notes, you should call it an insulin receptor. And so let's say insulin gets a high enough dose and it stimulates the cell. Okay, and the response would be there's these glucose transporters that will move to the cell surface to take the glucose out of the blood. So I mean, I don't want to get too fancy in how I draw it, but say there's a little glucose transporter. In response to insulin, it'll move to the surface and bind glucose. So the cell has a mechanism to take up glucose in response to insulin. So this happens after a large meal, right? A meal is an insulin trigger. So after your meal, you go sit down and watch television, let your food digest, and this happens hours after eating. Okay? And so ultimately, what have you done here? Because of insulin, you're going to lower the blood glucose, right? So that's the last thing that happens. Homeostasis examples are always good with the teeter-totter analogy thing, the seesaw thing, because if the imbalance is it's too high, just always tell yourself, oh, okay, I want to lower it. And the inverse is true. If the imbalance is blood glucose is too low, you want to raise it. So there's the same system to help raise glucose levels for glucagon. But I won't do that one. Just make a note to yourself. Maybe you study it on your own. You are responsible for glucagon. I'll get to the pancreas later. But I just wanted to give you a generic example of what you'll be studying. Every time you study something in endocrine, what are you trying to correct? What's the gland? What's the hormone? What does it affect? And what's the end result? You have to do that every single time. Another thing I'd like to do to introduce the chapter is the contrast, just the term endocrine with exocrine. Because they both come up in A&P, and I don't want you to get confused when you come across it. Endocrine, endo means within, because the hormones are in your bloodstream. If something's in your bloodstream, it's in you. By the way, the clock is wrong.
Yeah.
Schedule is wrong and the clock is wrong. It's a great first day for me. Oh, I'll get it straightened out. Okay, the endocrine glands, again, what I had said was they secrete hormones in the bloodstream. Okay. Now, an exocrine gland, it'll secrete it somewhere else, not in the blood. Usually on the skin surface or in the GI tract or something like that. And it's usually not, well, it's always not a hormone. It's always something else. So I call it non-hormones. It could be saliva, could be sweat, tears, most often in digestion is enzymes. Okay, just not hormones. And these endocrine glands, you need a duct to the surface. I'll just put, need a duct. I'll just put duct. They're glands that have a duct. Endocrine glands are ductless. They just need access to the blood. They don't need a duct that goes somewhere. Some differences there. And, well, they're both derived from epithelial tissue. I don't know if you remember studying that, chapter 4 of the book. But usually they look like cuboidal epithelia. ET is epithelial tissue. Okay, so they both kind of look like cuboidal epithelial tissue. Their function is endocrine, but they can all, okay, endocrine tissues can be epithelial in their derivation. They can also be nervous tissue. Neurons secrete hormones. Add that to the list. Because I'm trying to retrain your brain. In 430, we teach you how neurons secrete neurotransmitters, but they also secrete hormones. And we'll talk about that today. All right, moving on. Here's a picture of the formation of an endo versus exocrine gland. On the this picture here, you see that originally there's just a downgrowth. These cells have a glandular function. If it's an exocrine pathway, these cuboidal cells remain to form a duct, and the cells colored in red here have the secretory function. On the endocrine pathway, the duct cells just go away, and the endocrine gland cells have access to a capillary. Okay, that's what they're trying to show you, duct versus ductless. Now, when we look at endocrine glands under a microscope, we have this kind of array of things. You don't know what you're looking at now? I'll teach the histology of the different endocrine glands on Wednesday. So just kind of a preview for now. This is the different images we're going to have to look at. And all of the pictures that I take, that I test on, are actually from our slides. Not all of these are our slides, but what I usually do to present it is we have a camera here, and some people use it for 430, I do. I'll just kind of get the slides that you're going to study for lab. I'll take pictures of it. I'll present it up here so you can see what I'm looking at. I'm able to capture images, and I'll even use those images for your histology quiz, which I have scheduled for Friday. Okay, so that's kind of how I handle histology quizzes. I don't pull from the web. I like to think of it as a lab objective, which means I want to use the materials in our lab, in these four walls, and the slides are over there. Okay, so that's kind of how I'll handle it. I'll present histology on Wednesday. Moving on with the physiology, I want to talk about factors that affect the hormone finding their target. Factors affecting target cell specificity. Because if you secrete it way up here in the pituitary and you've got to find cells in the kidney, you can't react to everything along the way. You only react to things that have a receptor for you. Communication in the body is very specific. I'll just give you a simple example. You ever accidentally send a text to someone, but it was a group text and you forgot it was a group text? but you wanted that message only to go to that one person, but everyone saw it and you're kind of embarrassed because other people in the group, you weren't supposed to see that. The body's like that too. You secrete these hormones, you don't want everyone reacting to it. Only the specific tissues for the physiology you want to achieve. Okay, so specificity is very important for hormones. All right, I have a picture here. You should be able to appreciate the three fluid compartments. It's not necessarily a study guide point, but you have to appreciate that the hormones are secreted in the blood. Blood is contained within blood vessels. Blood vessels are containers for blood. So that makes it a compartment. So all the fluid outside blood vessels and outside cells is called the interstitium, or extracellular space, or extracellular fluid. Okay. The cell membrane, the lipid bilayer, creates a barrier. So that all the fluid inside cells is another fluid compartment. So that's three fluid compartments. And technically there's a fourth, lymphatic, but don't really worry about that for now. We're talking about the bloodstream. So the reason why I present this is hormones have to diffuse from here to here. Okay? And so factors that affect a hormone in blood getting, say this is a target cell you're trying to reach, a few things are outlined in your book. You have to secrete enough hormone. Call this factors affecting target cell specificity. Number one, you need enough hormone, okay? You need high enough hormone concentration in the blood. You don't need much. Hormones are very sensitive molecules. Books usually report anywhere from 10 to the negative 9th molarity to 10 to the negative 12 molarity is enough. That's not very much, but you need enough. If it's not enough, you'll never find a receptor on the target cell. Well, the other thing is the target cell has to present the appropriate number of hormone receptors. So cells will either upregulate or downregulate depending on upregulate target cells if, let me read right here, upregulation. Target cells will form more receptors in response to the hormone, in response to rising levels of the hormone. It'll present more receptors because to accommodate all the hormones circulating. Call that 2. Upregulate hormone receptors in response to rising hormone levels. The converse is a cell will start to downregulate the hormone receptor if the hormone levels are persistently too high. Okay, you don't want to overshoot homeostasis. Downregulate hormone receptors in response to persistently high hormone levels. A good example of this is diabetes, like type 2 diabetes, where if you're younger, healthy, and you secrete insulin after a meal, the blood sugar goes down. Your body responds to it. Over time, maybe due to poor lifestyle, poor diet, physical inactivity, you become resistant to insulin. It's not that you don't have insulin. You have it, it's just not working. That's called the type 2 diabetes. A lot of people think diabetes is a sugar problem. It's not. It's an insulin problem because insulin regulates the sugar. So it's like you eat a meal, your blood sugar goes up, your body secretes insulin, blood sugar doesn't go down. The pancreas doesn't know what's going on. So, oh, okay, the blood sugar stays up. I'm just going to keep secreting more and more and more and more. But because you're not sensitive to the hormone anymore, you have super high insulin along with the super high glucose levels because it's just not working. The cell, the muscles, they're not responding to the insulin. You just can't get it out of the bloodstream. All right, so the third thing I want to put is there a mutation in the hormone receptor? Is there high affinity? High affinity means it binds tightly. Okay, fits well, binds tightly. Hormone receptor affinity. You basically want high affinity. Okay, you'll get a response. Basically, in this situation here, you're not going to elicit a response in this situation. Okay? So hormones have molecular shape. They're either proteins or steroids. Some books give a third or fourth category. I basically boil it down to hormones can be molecularly proteins or steroids. That's what you're responsible for. Now let's remember what proteins are. They're composed of amino acids. Here's the general structure of an amino acid. There's a nice colorful graphic of all 20 amino acids. You don't have to memorize this table, but I do want to expect students to at least remember the basic structure of an amino acid. Protein hormones, review, amino acid structure. Basically, you got a carbon and it's surrounded at 4 positions by a hydrogen. The R group is also called the functional group, but there's always an amine group, nitrogen-containing amine group, and an acid. I would say the thing to know about protein hormones for this class is that they're water soluble, they're not lipid soluble. I'm going to put that somewhere here. Key thing to know about protein hormones. Fact I want you to know. Because in contrast, steroid hormones are different. Steroid hormones have this kind of hydrocarbon chain ring structure. more or less that. These 4 hydrocarbon rings. And steroids are classified as lipids. These are lipid soluble. They're not water soluble. I have a picture of cholesterol. Cholesterol is not a hormone, but all steroid hormones are derived from cholesterol. That's why I have a picture of it up there. One example could be the sex hormones, estrogen and testosterone. Okay, it's called, testosterone is the male king androgen. That's why they have a little crown there. Now, a third class of hormones, air quotes, I don't consider eicosanoids a hormone, so make a note to yourself, you're actually not responsible for this. I may mention it in another chapter. I think your book does mention it, but you can just kind of skip over it. You're not responsible for eicosanoids. Just stick to proteins and steroids. Okay. Now, the reason why we're sticking to those two is it kind of shows us how hormones affect target cells differently. Okay. Hormones are molecular triggers, but they're going to trigger the target cell in different ways. And here's how. So the basic idea here is that a steroid hormone, since it's lipid soluble, like dissolves like, so it can kind of go right through the cell membrane. It can even go right through the nuclear envelope and bind its receptor inside here. Okay. Protein hormones, because they're water soluble, they can't pass the hydrophobic zone of the lipid bilayer. So the cell has to present the surface receptor on the cell surface. So if this is the hormone and you want to get an effect out of this cell, you can't get through the cell membrane. You have to kind of knock on the door through the hormone receptor, and that'll trigger an intracellular response, usually second messengers. So protein hormones that act this way, they use second messengers. Steroid hormones, they just, it depends. Should draw a nucleus in both cells. A steroid hormone can find its receptor in the cytoplasm, or even in the nucleus. Because it's lipid soluble, it just goes right through and binds it intracellularly. And the thing about steroid hormones is they kind of call it direct gene activation because the hormone receptor complex turns genes on. Okay. Direct gene activation. Here's the professional picture of the direct gene activation. In this illustration, they show the steroid hormone binding its receptor in the cytoplasm, right there. And then it enters the nuclear envelope and binds the hormone response elements. You're going to get proteins. Okay? Transcription, translation, when you bind this promoter region, this hormone response element. Now, for second messengers, there's more steps, so I should probably take more time with this. But what we're seeing here is the hormone, which acts as the first messenger, it never enters the cell. It has to act through this receptor on the cell surface. Okay, so there's a G protein here. Now there's many different kinds of second messenger systems. If you take molecular biology, that's the class we learn all of them. But let's just learn this one as an example of you got to bind at the cell surface and use second messengers. So the first thing I have outlined is the hormone is the first receptor and it binds its receptor on the cell surface. You have a stimulated G protein that moves along the inside of the surface to activate adenylate cyclase. So I'll put G protein activated, moves, stimulates adenylate cyclase. The adenylate cyclase will convert ATP into cyclic AMP. You're just going to cleave off two terminal phosphates. Adenylate cyclase generates cyclic AMP. That's the second messenger. The cyclic AMP. What that will do in turn, the cyclic AMP, it'll activate kinase proteins, and that'll trigger more responses generically. Typically, I recognize cyclic AMP as the quote-unquote second messenger. Okay, but all these proteins and G proteins and adenylate cyclase things, they play a part. The kinase activity that comes after. So I'll put cyclic AMP increases kinase activity. That activity will get stuff done in the cell. Okay. The analogy has been given for like these second messenger systems because the communicating molecule, the hormone, it never enters the cell. But you got to get the cell to do stuff on the inside. So the analogy is given like you're walking along one evening and you see a house on fire. So you go pound on the door, trying to wake the people up inside so they don't die in the burning house. But you can only manage to wake up a small child who comes to the door, doesn't open the door because we train our kids, don't open the door to strangers, right? We should try to do that. My kids. And see, I can't open the door, stranger danger. But you're like shouting through the door, hey, your house is on fire, go wake up the parents. And so maybe they go do that and they take care of the fire. All right, so in my analogy, who's guy pounding on the door? The hormone, right? Does the hormone ever enter the cell? No. Remember that. This is like on the test, and sometimes students tell me how the hormone enters the cell, and at that point, you're sunk. Okay, it's like the child is like the hormone. Who's like the kid that goes wakes their parents up in this analogy? Yeah, something like the G proteins or the second messenger. The point is, the child is like these molecules acting intracellularly. The hormone was just the trigger, right? It didn't do all this work on the inside. It's just the trigger for all this. Okay. Now, one thing about this is I don't like how they're kind of generic examples of molecular biology, and they don't give you any physiology to learn. And this is a physiology class, so in the first unit, I like to use an example of a steroid hormone and a protein hormone that you have to know for this class. So as an example, let's learn the physiology of ADH and aldosterone. Let's remember that hormones are always trying to correct something. In this case, maybe you're dehydrated and blood pressure is dropping. So to kind of help conserve water for the body, the kidneys can help. The kidneys are constantly filtering your blood plasma, and to conserve water, what happens is the posterior pituitary is the gland that kind of senses the dehydration, and I'll teach the physiology of that starting today actually. But dehydration will cause posterior pituitary. It'll secrete the hormone ADH, which stands for anti-diuretic hormone. A diuretic, say for example alcohol, is something that increases your urine output. If you're trying to correct dehydration, do you want to increase urine output? Yes or no? Think about that. No, because isn't increased urine output, you're losing fluids, but you're dehydrated, you don't want to lose more fluids, so you don't want diuresis. You want a hormone that does the opposite, anti, right? This hormone is also called vasopressin, also known as, I'll say AKA, vasopressin. I'm kind of very big on the also known as. I try to give students as much ammunition so wherever you go next, hopefully it's something you've heard, okay? Whenever you see the term pressin in physiology, that means increased blood pressure, okay? So you're increasing the blood pressure. So one way you can do it is shown here. You got a kidney cell. And on one side, they call it the filtrate, colored yellow. That's basically the filtered blood. And you modify the filtrate. It will become the urine, okay, when it ends up in the toilet. But along the way, the cells will kind of conserve and take things out of the filtrate so you don't lose it to the urine. So on the other side, you have a blood vessel. I guess one thing to point out that I've noticed over the years is the osmotic gradient. 300, 600, 700. Milliosmolarity, the concentration. Water tends to go to where the more stuff is to try to balance it out and dilute everything. However, everything's watertight in this picture here. Water can't easily seep through the cells. There's tight junctions there. So, but the cell itself can regulate the water reabsorption. So the goal is, so you don't lose all this water to the urine, you want to reabsorb it back into the bloodstream. So the first thing that happens is dehydration, you get ADH. ADH binds its receptor on the cell membrane here. You write that down. Now look at the picture. Is it binding it on the cell surface or not? Yes or no? Cell surface. Yes, cell surface. So does that mean it's a protein or a steroid? It's a protein. Okay, it binds on the surface. It's going to increase the second messenger signal. I'll just put up arrow cyclic AMP. ADH never enters the cell. Now, in response to that, you want to get all the water out of the filtrate. On the other side of the cell where the filtrate is, these storage vesicles contain ADH. So what you're seeing there is okay, so what you're looking at there is, draw a little bigger. That little blue thing is a water pore, aquaporin. The rest of the black thing that they illustrate is the quote-unquote storage vesicle. You want to take that aquaporin and you want to stick it in the other side. Okay, say maybe right there. Just get it like aquaporin, AQP as an abbreviation. Get it there. Because once you do that, you stick it. Maybe another one on the other side. Now you have a channel for water to go through. Water would just flow down its osmosis gradient, right? So put H2O. So what we call this to move the water from the filtrate to the blood, call it water reabsorption. That's the physiology term, reabsorb. So you drink water, you first absorb it in your intestinal capillaries. Then the kidney filters it, and you reabsorb it, okay? And you don't lose it to the urine. And that'll help. Well, basically, it'll correct the dehydration, and when you add volume to blood, increasing the blood volume helps maintain the blood pressure if you're dehydrated. Okay, so increased cyclic AMP, you insert this water pore, they call it the apical membrane. So it goes apical, the other side is like basolateral, that's usually the terms that are used. Insert aquaporin to on apical membrane. Number 4, water reabsorption. Via osmosis. When you increase blood volume as a result of this, you increase blood pressure. So that's the end. Like, drum that in your head. Increase blood volume, increase blood pressure. It's a big theme in this class. Because it'll come up again for cardio. And it'll come up again a third time when we get to the kidney, because this is actually happening inside the kidney. Okay. So the hormone was ADH, came from the pituitary. It targeted kidney cell. I want to move on to the aldosterone example, but are there any questions on ADH? Here's some common mistakes. A student tells me on the exam, vasopressin enters the cell. Why is that incorrect? It never enters. Not even one time. It acts through a second messenger. Okay. Also, I would say if you fail to mention aquaporin, that's a big mistake. That's everything. Okay, it's like it doesn't get reabsorbed without the aquaporins. Okay. And it's very important that you connect the dots. I said this all occurs in the face of dehydration. So you got to tell me what corrects it. That one. Okay. All right, so moving on. This is also in the kidney, but a different hormone. This is a steroid hormone called aldosterone. It comes from a place called the adrenal cortex. I'll teach that probably Wednesday. Anyways, it sits right on top of your kidney. If you don't know where your kidneys are, it's like in your back, 12th rib. That's how I find your cadavers. Yeah, when the center of your back body, while the adrenal gland is on top of the kidney. And the cortex is the outer part of the gland, and that they secrete all these very important steroid hormones. This one regulates salt. So again, in the face of dehydration, same kind of trigger. Something I didn't mention before, you've usually also depleted your electrolytes, like sodium. So to correct that, a gland called the adrenal cortex, I don't get too ahead of myself, but there's a particular zone in that cortex called the zona glomerulosa, the cells that secrete aldosterone. Zona glomerulosa. Secrete aldosterone. That's where the figure comes in. The orange balls are aldosterone, and it's binding its receptor in the cytoplasm. Step 1. Aldosterone binds cytoplasmic receptor. Step 2. The hormone receptor complex, it turns on transcription in the nucleus. Remember, direct gene activation. You're turning genes on. I'll just say, turn on transcription. New protein channels and pumps are made. Okay, those are the things that'll help you recover sodium from the filtrate, help recover your electrolytes. Make new sodium channels, pumps. I know we use Na as an abbreviation for sodium. Na is short for natriuresis. And basically this is a word that means blood pressure. Yeah. Good old BP. Sodium helps maintain blood pressure, right? Hence the term Na for sodium. Okay, so you got your pumps there. Basically, they're showing you exchange a sodium that's reabsorbed for a potassium. Just forget about the potassium. Just know you get sodium reabsorption. You get that. I'm writing facilitates sodium reabsorption. Now the thing the figure doesn't tell you that I want you to know, water always follows sodium. Okay? It has to. So you get water with the sodium, because sodium is an ion that has a full charge. To be soluble in water, it's surrounded by, partial negative charges of water. So if you take sodium, water is following it. And, well, anyways, this will actually do two things. You'll recover the sodium, okay, of your lost electrolytes, and you're going to increase blood volume, increase blood pressure as a result of this. Increase blood volume, increase blood pressure. So ADH and aldosterone, they accomplish the same things, except with the aldosterone you also get the sodium reabsorption. But you're getting an increase in blood volume and blood pressure as well. Okay, I'll take a real break this time, not just defined in a bathroom break. The way I give breaks is I typically give a 15-minute break halfway through the lecture, and I clear out the room. I lock the door. I don't let anyone stay in. That way I'm allowed to leave and I can get a cup of coffee somewhere. So go ahead, take whatever you need. I'm going to lock the door and come back in with your watch, 15 minutes.
I know you're going to fix it later. But on the
Yeah, all 3 holidays are off.
Okay.
I got to fix them all.
Okay.
Yeah, completely botched it.